It's a pleasure for me to introduce Dr. Pawel Ciborowski. Dr. Ciborowski is the director of the Mass Spectrometry and Proteomic Core Facility at the University of Nebraska Medical Center, and is a professor, assistant professor in the departments of pharmacology and biochemistry. Dr. Ciborowski has been collaborating with us in the neuro-AIDS field, and he is, actually Dr. Ciborowski is native from Poland, and he graduated from Master in Biochemistry and finished his PhD in Microbiology and Biochemistry at the National Institute of Hygiene in Warsaw, Poland. He subsequently performed 2 years of postdoctoral studies at University of Cologne in Germany, and also became assistant professor there in Poland. And he joined at the University of Nebraska Medical Center in 2003, and since then has been a collaborator in the NeuroAIDS program. He has more than 20 publications in the field, and he will talk about HIV-associated cognitive impairment and proteomic approach. So, Dr. Ciborowski.
So first of all, I would like to thank the organizers for inviting me here. It's, I think it's my 9th visit here in Puerto Rico, but it's always a pleasure to come and to talk about our results. So today I will be speaking about HIV-associated cognitive impairment and a proteomics approach that we apply to study this disease. HIV-associated cognitive impairment, there are also other definitions of that disease, of this pathological state or disease, that is dementia complex, ADC, also known as HIV dementia, HIV encephalopathy, and HIV-associated dementia, and so on, and as well as classification of this disease of a late stage characteristic for the late stage of AIDS is changing as we acquire more knowledge about it. It is not very simple to classify and to diagnose because of two major reasons. One is that dementia or associated with AIDS could be caused by many different factors, such as opportunistic infections, cerebral lymphoma, or metastasis of other AIDS-related cancers, direct or indirect effects of HIV in the brain, the effects of drug treatment, as well as drug and alcohol abuse, and malnutrition of people suffering from that. So there is no really laboratory test that can discriminate between HIV-associated cognitive impairment from the other reasons that can distinguish the factors that are also influence. Diagnosis right now is based on the psychological and psychiatric tests, and there were many attempts to find biomarkers as well as correlate parameters such as viral load, CD4 count, and so on. However, none of them were really good discriminatory factors. So what is the consequence of this disease? It is rapidly progressing and is ultimately leading to death. There is a lack of correlation or association with race, gender, or age. And what has been observed was that the HIV-associated cognitive impairment or HIV-associated dementia is correlated with infiltration of central nervous system with macrophage and with activation of a macrophage. So, of course, the first question is there any correlation with HAART therapy, a highly active antiviral therapy? And surprisingly, although HAART therapy really extended the life of infected people, it had minimal or maybe no effect on the rate of the dementia. And even one of the latest review papers co-authored by Avinat from Johns Hopkins is saying that HIV-associated cognitive impairment is on the rise, and we expect even more cases to come. Now, why macrophage is important? Macrophage is important because macrophage, unlike a T cell, can coexist with the virus. So when virus infects T cell, it kills it. Now, when it infects macrophage, and that will be macrophage tropic strains of HIV, can migrate to the different tissues as well as going to the central nervous system. And one of hypotheses that I will show you here in this slide is that actually macrophage is bringing a virus to the central nervous system through the blood-brain barrier. There are also other hypotheses of a direct passage or the passage through the endothelial cells. However, the hypothesis of macrophage or monocytes infected on the periphery and carrying a virus to the central nervous system is the most prominent right now. Now, once going through the blood-brain barrier, a virus is further disseminated, and because there are 4 different potential outcomes of infection, is that infection could be either abortive, that could be latent, permissive, or restrictive. And if this is permissive, then there is a viral production in the central nervous system, and the virus can further spread to and infect other cells of a central nervous system, such as astrocytes. Whether pericytes or neurons are infected, it still is the matter of debate. So our hypothesis was that the serum and cerebrospinal fluids will contain a spectrum of mononuclear phagocyte activation markers or fingerprints that we can use to predict HIV-associated dementia. So we used proteomic approach to study this problem, and we did that in 3 steps. Step number 1 was that we look at the secretome of in vitro HIV-infected human monocyte-derived macrophages, and then which we completed, and we recently published a paper reporting results from that study. And then in the second step, we did a proteomic analysis of cerebrospinal fluids from HIV-infected individuals with or without cognitive impairment. And this study is completed in a way that the manuscript is right now submitted and being under revision. Here I shaded that intentionally, the step 3, which is in progress, that this is proteomic analysis of sera from HIV-infected individuals with or without cognitive impairment. And we are looking also at potential biomarkers in serum or in plasma. We have completed the first step. However, we have not validated results yet. That's what I will not be showing them today. However, it is important to remember that there is a communication between cerebrospinal fluid and blood, so we also expect that some markers can be transferred from one fluid to the other. So in the first step, we use serum-free culture supernatants of HIV-infected human macrophages, and then because this is relatively simple a sample, I don't know how many proteins, but we think that it might be around 700, 800 proteins versus more than 3,000 in the cell lysates. That's why we use the simple one-dimensional SDS page as a prefractionation, and I will show these data in a moment. And then we selected potentially differentially expressed proteins, and then we validated them by Western blot analysis. So here is, it shows the typical culture as well as the assay of reverse transcriptase, which shows that there is a productive infection going on in vitro culture. This is a control, monocyte-derived macrophages, and here we can see that there is a progressing infection, which is characteristic in the way that macrophage creates syncytia, which are also called multinucleated giant cells. In another forum, a journal, virology journal, like that so much that put these pictures on cover. So here is our experimental design, very simple. We have control supernatants, we have HIV supernatant from HIV-infected macrophage. However, as we can see, that despite of the fact that we grew for the last 24 hours prior to collection, we cultured macrophages in a serum-free medium, we still have substantial amount of serum as well as IgG, which is heavy chain and light chain. So for that particular region, what we did, we removed immunodepleted culture supernatants from albumin, and as we can see that there were some additional proteins which were not present in a control culture supernatant, but it were present in a supernatant of HIV-infected macrophage. So a proteomic study usually yield a massive amount of data, and showing that it wouldn't be possible here. So I showed a summary of combined several experiments that we have done, and the identification for this, for everything that I'm presenting today, was done using nanospray LC-MS-MS, and we have LCQ Deca Plus, and we use sequence algorithm for searches. Here are our criteria that we use X correlation factor more than 2.5 for doubly charged species, delta CN 0.3. That's a little bit higher than other people use, but we felt comfortable with that, NSP greater than 500. Now, we divided our identifications in two groups, the high confidence and low confidence identifications. When two or more high confidence identification was when two or more unique peptides were sequenced with high confidence, and these are the numbers of the proteins. And here is the low confidence when one peptide of a high quality was sequenced, and we have additional proteins. So the total number of proteins identified were 261, and probably this is not all the proteins that were present. However, maybe there were some proteins of a very low abundance that we missed, which is very possible. So then we went into identification. From identification to validation using Western blot analysis. This is the time, the day after infection, so the first, second, third, 5, 7 days after infection. We have controls and we have infected. This is Western blot for HIV protein P24 that shows that indeed 5 days we were able to detect viral proteins, which confirm the RT assay showing the productive infection. And here we have a cystatin C, which is downregulated. This is chitinase 3-like-1 protein, which is also HCGP39 protein that is slightly downregulated in HIV infected. But cophylline represents also structural proteins which are not present in an uninfected or in a very early stage of infection, but they are present in HIV, in culture supernatants of HIV-infected macrophage. Here we have upregulated L-plastine, and in the next panel shows again that upregulation of superoxide dismutase, which was very surprising because we thought that we kind of expected that SOD will be downregulated during infection, and that may contribute to the damage done to the brain. Here is another leukotriene A4 hydrolase, alpha enolase, and cystatin B, which is highly upregulated in HIV infected macrophage. So here we have cystatins. The reason I'm focusing on cystatins because this is the subject of further investigation, and together with Loida, here we are preparing a proposal to be submitted as a NIH grant. And it is interesting because cystatin B, which is upregulated, is the intracell is an inhibitor of catepsins, while cystatin C, which is downregulated, is inhibitor of cysteine proteinases. So in other words, one inhibitor goes up, one inhibitor goes down. There is a substantial work done on the role of cystatins in other diseases such as rheumatoid arthritis and how they are or potential role they play during the rheumatoid arthritis and synovial fluid and so on. However, we know very little, if any, about the role of cystatins during the inflammation of a central nervous system. And of course that we are interested in the biological meaning of that, what we are finding. So after we completed that step, we proceeded to the next step, which is the proteomic analysis of cerebrospinal fluids, and with the plan that if we can combine the results from in vitro studies and ex vivo studies, we will find the common proteins as to support our hypothesis that indeed HIV-infected macrophage is contributed or can give us biomarkers of this neurodegenerative disorder. So first of all, I would like to briefly discuss the challenges in studies diseases of central nervous system. First of all, cerebrospinal fluid is only material which has contact with the central nervous system and can be obtained from living individual. Unfortunately, we cannot obtain any biopsies or any other material of that tissue. There are intrinsic problems in proteomic studies on the brains obtained post-mortem through the rapid autopsy program. And why is that? Well, the recent lecture that Bruce Brunson gave in Nebraska showed that that hypoxia or lack of oxygen causes changes in the white matter in a matter of seconds and minutes. So the few hours that it takes to retrieve the brain from the diseased that person may have already changes which will be masking the real biomarkers. Now, protein concentration in cerebrospinal fluid is 10 to 100 times lower than in serum, and only up to 20 ml of CSF can be drawn from individual at one time point. Therefore, the size of sample is limited and not readily available. Now, the fact is that what I found somewhere that this is legal to draw up to 20 milliliters, but it doesn't mean that always 20 milliliters is drawn from a patient. And even if 10 or 15 milliliters of a CSF is drawn from a patient, it doesn't mean that we researchers get all the sample. So the size of usually the size of a sample that we get is around 1 milliliter for the study. So here is how we process CSF samples. So first of all, we have to add Triton X100 to the samples because these samples are from HIV-infected people, and as such, even if there is a zero viral load, they are considered to be infected with the live virus, and therefore they have to be handled in a biolevel safety laboratory 3, and the virus has to be neutralized before we take these samples out of a BSL-3 laboratory. And we also add protease inhibitors. Some people say, well, it is not really necessary because there are natural inhibitors of proteases in the sample, but we know also from other studies that it's really important. So we use the cocktail specifically from Sigma. Now, then we remove immunodeplete these samples from 6 most abundant proteins, which is albumin, IgG, IgA, transferrin, haptoglobin, and antitrypsin. And we use that Agilent Technologies column Q6. We collect the samples, then we precipitate using 2D cleanup, a kit from GE Healthcare because we need to reduce volume, as well as we have to reduce Triton X100 because during the concentration, Triton X100 will create micelles and will be heavily interfere with the mass spectrometry analysis. So here then we do the protein determination, and we label them with fluorescent dyes, site 3, site 5, and site 2, run a 2-dimensional electrophoresis, do image analysis, spot picking in gel tryptic digestion, and LC-MS protein identification. So here is the range of the samples of the protein in the CSF samples. So here we measured the protein concentration in 40 CSF samples. And as you can see that we have as low as 160 micrograms of protein per 1 ml. Those two, I think, that might be outliers, but an average is a half milligram per ml. So when we obtain 1 ml of a protein, 1 ml of protein, that is all what we have to live with. Now, this is what we expect after immunodepletion, because after immunodepletion, we think that 90% of protein is removed, so we can end up with as little as 16 micrograms of protein total. And the next slide shows another variability, which is another challenge in analysis of proteomic analysis of cerebrospinal fluids. Is we did that with the four samples from HIV demented people and 4 from non-demented, both groups HIV infected. This is how much we used in micrograms before immunodepletion, and here we have that after recovery, after immunodepletion, and the yield in percent. And what is striking here is that in a group of non-demented individuals, we have maybe between 21 and 12%. That's less than a twofold difference. So this group has a pretty uniform ratio of the 6 most abundant proteins to the other proteins. However, when we look at cerebrospinal fluids of HIV demented people, as we can see here, that in one individual gave us only 8.3% of a protein, total protein after immunodepletion. In the other one gave 35%. So the yield of after immunodepletion is really very diverse. And this is important. The reason I'm showing this is that this is important for normalization and for interpretation of our data. So here is the schematic representation of our approach when we have control samples which this is a little bit misleading. Control will be a non-demented and infected will be demented. So they are labeled with Psi3, green fluorescent dye, and Psi5. And here, when we mix them together, run on 2D gel. If we have both, we should see more yellow. If we have only in control, that will be green, and only in infected or demented, it will be red. And here is one of our typical gels. Okay. Now there are these circles. They show some proteins that we initially look at and try to identify. I will speak a little bit about this red one, which obviously everybody will ask, okay, so you have the strong protein expressed or present in HIV demented individuals. Now, there are individual samples, and I would like to draw your attention to the last one, which is green, which was indicating that this is a non-demented individuals. However, this is carboxymonohemoglobin identified. Now, what it tells us is that probably during the lumbar puncture, spinal tap, the little vessel was poked, and then the sample will be contaminated with blood. And this is very important, and I agree 100% with Dr. Costello, who said in a comment about the challenges in proteomics is the quality of samples. Because now if we can, if this one sample is contaminated with blood, that will be also contaminated with plasma, and then interpretation of this data might be misleading. So this is another challenge. But here I would like to go to that hot red spot, which we identified with the in both cases. I'm showing only 2 gels for the clarity of presentation, but we did it multiple times, and in each spot we found 8 peptides, 8 unique peptides for vitamin D binding protein, as well as a molecular weight and migration of the spot. Also, isoelectric point were corresponding. So we are confident that really these spots, they represent this protein. Now, unfortunately, we were unable to validate this data using Western blot analysis, and we don't understand why. Why was that? Maybe this was a matter of antibodies. I remember listening to the presentation by Jennifer Van Eyck from Johns Hopkins, and she said that to really go from, when she was presenting her data from PF to D analysis and then confirmation by Western blot analysis, she said that in many instances she has to use many or try many antibodies to find the right one that will confirm. Now, why this protein might be interesting? Well, this protein has a single site of O-glycosylation. It is mucin type, so this is GalNAc-Gal sialic acid, and on the side it has a fucose. And when this oligosaccharide is trimmed down to T and antigen, which is GalNAc linked to the serine or threonine, then it turns this protein in a very potent macrophage activating factor, which is considered to work in a way as a cytokine. So we were also very excited about that. However, using lectins, specific lectins from Helix pomacia, we were unable to confirm that or find any differences. This time, the reason was that it was co-migrating with a very strong band with another protein, which was very abundant and had a lot of T and antigen. So limitation of the size of a sample pushed us into the 2D Dij analysis with a saturated labeling. When we used samples from non-demented as well as from demented individuals, they were both labeled with the Cy5 red fluorescent label, and the mixture of all the samples they were labeled with Cy3, and then we ran 6 separate gels for 1 sample each, but we had equal amount of the internal control for quantitative analysis. At the same time, we ask another question: is how many samples we need to run to obtain results and to find the differences. We know that, first of all, this technique is very laborious, is long, as well as it is costly due to the high cost of fluorescent dyes. Now, here the advantage of this approach is that, that for a preparative gel, we can pool the remaining portions of the CSF, which will increase our chance to identify proteins, medium to low abundant proteins, by mass spectrometry. and we can load as much as 500 micrograms of protein, which will provide enough identification. Okay, and for the analytical purposes, we can use as little as 7 micrograms. Of course, that we will, if there is a limited sample, we will not have enough for the Western blot analysis. So here is just an example of two gels from non-demented and from a demented individual. And now I'm going to show three examples of decider analysis. We use a decider software from Amersham, formerly Amersham, and which gives us, even if we have only 3 in one group, still we can calculate the significance. And here it shows that indeed we have the significant difference. Here we have a spot that is consistently decreased in HIV demented, although with a statistical significance. And in a third example, we have 2 samples very low, 2 samples very high. However, there is 1 sample in each group that is in the middle, and that creates a problem with statistical significance, which is a little bit above 0.05 in the t-test. And here there are images of the corresponding spots. So here, that after we did statistical significance and the analysis of these samples, we automatically analyzed 3,878 spots in all 6 gels. Out of these spots, only 1,350 spots met the following 2 criteria, which were detected in at least 2 out of 3 gels in one group, and their positions or coordinates were matched in all gels were detected. Now, out of these 1,350 spots, 90 spots showed difference between groups with statistical significance of p-value lower than 0.05, and another 14 spots showed the difference with the p-value in between 0.05 and 0.06, which we considered that it might be if more samples are tested, that will be more, that may be significant difference. Now, here, this slide shows those spots that we selected for further identification using nanospray, nanoelectrospray, LCMS-MS. And here is the table summarizing our data, where we have a Deitch index, and if the negative value for the Deitch index means that this is down-regulated or lower level in demented in samples from patients with HIV-associated dementia, and they are sorted by a Deitch index. Here we have a p-value showing the statistical significance, identified protein, as well as isoelectric point, and so on, and accession numbers. Now, what we can see is that we have complement C3. We have the same protein identified in multiple spots. However, in all the spots, this protein was downregulated. Here also we have spots in which we identified 2 proteins with a high confidence according to the criteria that I mentioned before, and interpretation, or here we have 3 proteins even in one spot. So that's why interpretation of these data whether which proteins, one or maybe all or 2 out of 3 were contributing for the -3 or almost for Deitch index. And the second part of this table shows that we go from the -8 to the +3.4 when we have some proteins like histocompatibility antigen HLA, but also prealbumin was increased in HIV demented individuals. Here we have cystatin C, and cystatin C was present in the 3 spots, and here it was almost 1. So it was not really consistent with our data shown by in vitro studies, because we showed that cystatin C is downregulated. But I will come back to that issue in a moment. So here is validation. Validation is important, and there are different methods of validation in proteomics studies, and I think that everybody who is doing proteomics is to some extent struggling with that, and we choose to validate our data by Western blot analysis. So here is a summary of Western blots, one representative spot from the CIDR analysis, and 4 samples. Now, we use for this portion of validation, we use different samples that we used for the biomarker discovery phase. And as we can see here that, for example, that there is Joseline, which is downregulated, clusterin is downregulated. On the other hand, if we look at these data, we can see that even within the same group, there is a high diversity of the level of a specific protein. But when we look at the complement C3, we can see here that in non-demented, this chain, this form of complement C3 is consistently expressed. However, in demented, in the CSF samples from demented, this is consistently lower, even up to the negative. Some other, like procollagen C peptidase enhancer, we don't have that clear results like for complement. However, again, in demented individuals, there is a high diversity in the levels of this protein from relatively high to almost nothing. So here it shows that the complement is present in multiple spots, and we found that consistently here. We found all complement C3, and in all of these spots there was a decrease in HAT and an increase in non-demented. So our validation by Western blot and 2D analysis show consistent and reproducible results. Now, if we use for Western blot analysis, if we use those 3 samples that we used for discovery phase for 2D gel analysis, we can see that in non-demented we have high level of complement. In demented we have almost nothing in 2 patients and a little bit in the 3rd patient. So our approach, proteomic approach, it shows in some cases consistent results. This is again the same slide. Now I would like to go to another portion, and this is just to show you how the complement C3 is processed. That first there is a C3A fragment cleaved off, then there is a C3F, and then there is a cleaved C3DG. And what we have is this alpha 40 fragment, which is not really so exciting because it doesn't have any regulatory properties listed in literature. And in a processed C3 complement, it's linked with everything else by the disulfide bond. But when we apply reducing conditions, this fragment will be migrating on a gel separately. So here we have, we found that fragment on a 2D gel. There are two spots, and in both we found the same complement C3. And then how do we realize that this is alpha 40? Well, alpha 40 fragment is derived by the cleavage between 1320 and 1321 amino acid of a C3 complement chain, and we sequenced and we identified this protein consistently in all of these bands, as well as the other proteins. But in these two spots, we never found any other peptide from any other fragment of complement C3. And the next slide shows again that from independent experiments, this is the spectrum. This is from the BioWorks software sequenced algorithm of sequencing of this peptide, and this looks pretty reproducible. Now we could not confirm that and validate that and confirm that by Western blot analysis because there is no good antibody. Even the polyclonal rabbit antibody against the whole C3 complement does not react well or react very weakly with that alpha 40 chain. And we looked through entire world for the antibody that can give us better results. Here is again procollagen C endopeptidase enhancer. These are 3 samples that were used for the 2D analysis, so for the discovery phase in our proteomic analysis, and in non-demented, on average, that will be higher than in demented. However, one individual has the same level as the non-demented person. Here is another 8 patients for non-demented that we have more consistently showing the higher level of the samples in comparing to non-demented. This is a control that we used as an internal for densitometric measurements and to quantitate our data. So now the question is whether the Deitch index minus 135, which many people may question, that's minus 125. This is so little difference. However, this so little difference in a Deitch index, which is an average of 3 samples, really in this case translates to a difference that we can see in our validation phase. And currently what we're doing, we've got samples, a totally blinded a cohort of samples from the University of San Diego, CSF samples from demented and non-demented people who are running this test, and then after we classify them according to our test, we will see how well it matches with the medical or clinical classification. So now, of course, that we are doing that, we're doing all of these proteomic analysis because not only we would like to discover biomarkers, which is of course very desirable outcome, but also what we want to do is to learn more about the disease, and that's why we are more focused. And for the next step of our work is to try to get the sense of what these findings can mean in a biological sense. So, for example, again, I will give few examples due to the time limitation. It's this procollagen C endopeptidase enhancer. Proposed functions are, this is secreted protein, so that correlates that we found it in a cerebrospinal fluid, binds to the C-terminal propeptide of type 1 procollagen and enhances procollagen C proteinase activity, which may be involved in the regulation of matrix metalloproteinases, which we know that they play an important role during the brain inflammation as well as during the HIV-associated dementia. Because that may have the metalloproteinase inhibitory activity. Now, Joseline here is another example that in non-demented we have pretty much consistent. Again, in these 4 individuals, they were additional to those that we used in discovery phase, and that we have reduced levels with the Deitch index either 187 or minus 187 or minus 177, because we found Joseline in 2 different spots, and again is controlled for normalization. Now, Joseline is a interesting protein because there are 2 isoforms. One is secreted isoform, and the other one is the cytoplasmic isoform 2. They are different by only a short fragment of several amino acids from the N-terminus. Now, there is no antibody existing that can differentiate those 2 forms, cytoplasmic from secreted, as well as we were unable so far to find out which form we are detecting, because we were unable to get sequenced the N-terminus of this protein. Now, the function of secreted isoform 1 of Joseline is completely unknown. It is postulated that it is involved in some regulatory mechanisms, but I would say that this is more speculation and not really supported by any research. So here, going towards the end of my presentation today, we are looking where is the link between that what we found and that what the disease is. And here I brought also matrix metalloproteinase 9 that we published prior to that study, that this is that in vitro studies it is downregulated in macrophages. However, elevated levels are in CSF of HIV-infected individuals. So that creates, of course, the question, it's a contradiction, why the macrophage is downregulated while you find more. Now, the one explanation, I'm not saying that this is the best explanation is, but one explanation is that, first of all, during the inflammation, and certainly during the inflammation of the brain in HIV-infected individuals, more macrophage are recruited to the central nervous system. So not all macrophages are infected in the brain. So if we, so those infected, they may downregulate matrix metalloproteinase 9. However, the net value in the cerebrospinal fluid will be increased. That will bring us to the role of MMP9 in the local environment, and especially in the environment surrounding multinucleated giant cells, which I showed at the very beginning. Here, procollagen C endopeptidase enhancer that we found in CSF samples, but we did not find it in vitro studies, which tells us that that could be produced by cells other than macrophage in the central nervous system. There is no solid evidence of biological importance. Now, chitinase 3-like 1 protein, that HCGP39, again, we found it in vitro studies. We also tested cerebrospinal fluids for the presence, and results are ambiguous because the level of this protein is very, very low in CSF. However, what I learned from that other group of people who are doing infections of monkeys with HIV virus, and they were studying the cerebrospinal fluids, they also found that protein upregulated in CSF samples of these monkeys. Now, why this protein might be important? Well, first of all, the receptor for this protein is unknown. However, there is one paper showing that the binding of this protein to the surface receptor will cause the inhibition of the action of two pro-inflammatory cytokines, IL-1 beta and TGF alpha. So again, that protein may play a role in the regulation of the pro-inflammatory response of macrophage in the central nervous system. So here we have conclusions that we successfully used our proteomic studies in disease proteomics, that we have identified 261 proteins in culture supernatants, secretomes of HIV-infected and control MDMs, which can be used for future focused studies such as CGP39. Well, 2D-SDS page DIGE with saturated labeling was successfully applied to discover difference in CSF samples from HIV-infected patients without or with cognitive impairment using limited number of samples and limited amount of protein from each sample. Unfortunately, studying CSF samples, we do not have a luxury of 1 milligram of protein like we can get it from, let's say, from other tissues, or we can get from in vitro cell cultures. And using 2D-SDS page DIGE, we identified 29 differentially expressed protein spots with statistical significance representing 21 proteins. Of course, everybody may ask, would you expect more or would you expect less? Well, I don't know. Probably there will be more, but this is the outcome of the first round of this project. And initial experiments showed validation of discovered proteins on the separate cohort of CSF samples. And I admit that discovery phase was done on 6 samples. The first step of validation was done on 8 samples, so this is altogether 14, but as I said, with the collaboration with the University of San Diego and the NeuroAIDS program there, we are now testing a cohort of 40 samples. So we will be further validating our data. And then I would like to acknowledge people. First of all, Dr. Howard Gendelman, who is a director of the Center for Neuroviral and Neurodegenerative Disorders, as well as he is a chair of the Department of Pharmacology and Experimental Neuroscience, who recruited me 4 years ago to Nebraska to build and run the proteomics program. Wojtek Rożek, he is my postdoc, that he did most of the work for the CSF analysis, and other people who contributed to both of the projects, as well as I would like to acknowledge University of Puerto Rico Medical Sciences Center, Dr. Loida Melendez, Dr. Valerie Vojna, and Dr. Juliana Perez-Laspure, our collaborators. They supplied us with CSF samples from the cohort of Hispanic women with cognitive impairment. I also would like to acknowledge National NeuroAIDS Tissue Consortium that gave us sera and CSF samples, this program is supported by National Institutes of Health, as well as National Institutes of Health supported in parts of the work that I presented today. And with this, I would like to conclude my presentation. I will be happy to answer any questions.